Hi, my name is Jonathan. I'm an assistant professor here at the Center for Volatile Interactions, and I'm a microbial ecologist, and basically I'm interested in how the ecology or the dynamics in microbial communities affect biogeochemical cycles and greenhouse gas emissions. So I've been always in touch a lot like with outdoor activities in nature, particularly going to the mountains. But career-wise, I took a bit of a detour. So I studied biology with a focus on biomedicine. So I did my master thesis on brain tumors. But I felt a bit like the research was a bit disconnected from what is happening actually with the people. And then I started searching, okay, what else can I do? What is interesting to me? So I was like, I'm really interested in the environment, and I had this background in molecular biology. So I was like, okay, microbiology, that is like where the two come together. So I did 2 internships then to get a bit of a background. So one was in Aarhus in Denmark with marine environments. And then I came to Zurich, where I did another internship with soil and warming in alpine environments. And that was like where I really felt connected and like, yeah, this is really something I want to do. And so this is where I also did my PhD then. So basically, since my PhD, my work focused on understanding the impacts of climate change on soil microorganisms and the role that microbial communities play in mediating feedback to climate change, like for CO2 emissions. And basically a large part of my research made use of natural climatic gradients. For example, in my PhD, I worked in the Swiss Alps, but then also in Arctic environments in northern Sweden. So one thing is to use these natural gradients, and another thing I have done both in my PhD and postdoc projects is translocating soils, like for example from a colder place to a warmer place, and see how the microbial communities change and how this translates like into soil functioning and greenhouse gas emissions. And it was pretty cool. For example, with these projects, I could also go to Greenland and South Africa and see very beautiful places. So basically, to address microbial processes in the different climate experiments I do, we're looking at greenhouse gas emissions, for example, CO2, where you basically place a chamber on the soil, then the gases are coming up in the chamber and it's connected to a machine that records the emissions. We also take soil samples back to the lab and we measure different properties of the soil, like the pH and the texture. We also measure the climatic property, of course, like temperature and the water content of the soils. To look at the microbial communities, I use DNA sequencing techniques. So the problem with microbes is like there's many different microbes, and most of them we can't culture. So we cannot observe them live, let's say. So what we do basically, we use DNA sequencing techniques and match what we find to a database. So that can give us information what is going on in the environment. And so this way we can link the ecology of the microorganisms to the processes that are happening in the soil. For assessing microbial communities through DNA sequencing, there's basically 2 approaches. So one is marker gene sequencing, which basically shows the overall patterns in the communities, and then we can annotate it with a database which gives information about the taxonomic identity. So what is the name of the taxon? But sometimes it's not so much more than a name. And the other approach is called metagenomics. So we sequence all the DNA that is in a soil sample, and then we look at functional genes. So that can give more information about the function. But still it's quite complex because the whole process depends on databases, and they are not necessarily made for ecological interpretations. So sometimes you get information or categories like protein metabolism, which is like very generic, so it's hard to make a meaning out of this. And it's, for example, something I find quite frustrating during my PhD, because I felt a lot of the studies were very descriptive, and it was hard actually linking the microbes to the processes. So in my last project I had one of these data sets with marker genes. So it was derived from the data set I talked about before, so different climatic gradients in different countries. And I developed an approach for marker gene sequencing where instead of the taxonomic identity, it is possible to annotate the sequences with information about the traits. Like, for example, how fast do the microbes grow, how big are the genomes. So from that we can infer, for example, are they very specialized? Are they more generalist? Are they able to do a lot of different things? And also, are they able to withstand stress? Like, for example, are they resistant to high salinity or high temperature? And like with this kind of information, it's really possible to talk about ecological strategies. Like, we can learn how do the microbes adapt to a specific environment. One thing that is very interesting about my recent research is that I was able to pinpoint microbial strategies to adapt to specific environments. So we used the setup of different gradients across countries, measured the environmental conditions like the climate and the properties of the soil, and then inferred traits of the microbes from sequence analysis. These are traits like, for example, how fast do the microbes grow, how big is the genome. So we were able to identify certain strategies that enables the microbes, like, for example, to thrive in a desert, like in a very cold place, in a very hot place. And how opposed to that do they live in a forest soil where there's plenty of resources, where there's a moderate climate. So the next step is sort of to turn the whole thing around, like instead of asking how does the environment influence the microorganisms, use the microbial traits to predict the responses of the soils to the environment and link it back to greenhouse gas emissions. I recently started a new project. So the aim of this project is to develop a microbial model of VOC emissions. So VOCs are volatile organic compounds, and they are emitted from all kinds of living organisms and can affect the climate. And I work together with ecosystem modelers who look at VOC emissions from plants, but what we don't understand very well is the role of microbes in this process. And so I'm aiming to develop a mechanistic models of how the microbes affect VOC emissions. And yeah, I'm very excited to work together with the modelers and empirical scientists at Volt. Thanks for watching. I hope you enjoyed the video, and feel free to check out other videos on Volt.